Yes,
Am I audible? Huh?
yes, yes, yes.
Acha. So, a very good evening and a rousing welcome to today's webinar, which is organized by the Department of Chemistry in collaboration with the IQAC of Fakirchat College, Diamond Harbour. I am Dr. Proggamay Pal, Associate Professor and Head of the Department Chemistry, and also the present coordinator of the IQAC of this college. Our IQAC has taken a program of knowledge development through a series of events, and as IQAC coordinator, I am much glad that all of our departments have responded very enthusiastically in this matter. Today's program is a part of it. As you all know, today's topic of discussion is environmental hazards of polymers and its remedies. As it is one of the burning environmental issues of the day, so we have planned to organize this program on 5th June to celebrate the World Environment Day. But due to some reasons, we have to postpone it for more than a month. So, with these few words, I like to request our principal sir, Dr. Shomen Chando, to deliver his inaugural and welcome address. But before that, my earnest request is to keep your camera and microphone off. And also, after Professor Shunmugam's talk, there will be a brief interactive session where you can ask questions to our resource person. So now, Principal Sir, please you deliver your inaugural address, sir.
Thank you, Progga. Am I audible?
Yes.
Okay. A very good afternoon. Distinguished guests, speaker, dear colleagues, students, and ladies and gentlemen. Today's webinar titled Environmental Hazards of Polymers and its Remedies has been arranged by the Department of Chemistry in collaboration with the IPC of our college. As you have all heard from the head of the Department of Chemistry that this webinar has been arranged in commemoration of the World Environment Day. The environment has become a major concern for the civilization in general. Thanks to the organizer. I am not a person from the field of chemistry, so please spare me if I make any mistake in my short introductory talk, which is of general nature, and I hope that the students will be encouraged to study this kind of subject in future. Polymers are a class of natural or synthetic substances composed of very large molecules called macromolecules that are particles of simpler chemical units called monomers. Polymers make up many of the materials in living organisms, including, for example, proteins, cellulose, nucleic acids, etc. Moreover, you can see the basis of such materials as diamond, quartz, feldspar, and such man-made materials as concrete, glass, paper, plastics, and rubbers. The word polymer designates an unspecified number of monomer units. When the number of monomers is very large, the compound is sometimes called a high polymer. Polymers are not restricted to monomers of the same chemical composition or molecular weight and structure. Some natural polymers are composed of one kind of monomer. Most natural and synthetic polymers, however, are made up of two or more different types of monomers. Such polymers are known as copolymers. Organic polymers play a crucial role in living things, providing basic structural materials and participating in vital life processes. For example, the solid parts of all plants are made up of polymers. These include cellulose, lignin, and various resins. Cellulose is a polyacrylate, a polymer that is composed of sugar monomer. Lignin consists of a complicated three-dimensional network of polymers. Food resins are polymers of a simple hydrocarbon, isoprene. Another familiar isoprene polymer is rubber. If one hydrogen atom in ethylene is replaced by a chlorine atom, vinyl chloride is produced. These polymerizes to polyvinyl chloride is a colorless, hard, tough thermoplastic material that can be manufactured in a number of forms, including foams, films, and fibers. Vinyl acetate, produced by the reaction of ethylene and acetic acid, polymerizes to amorphous, soft resins uses, used as coatings and adhesives. It copolymerizes with vinyl chloride to produce a large family of thermoplastic materials. The non-biodegradable nature of synthetic polymers make them a permanent waste. The used polymeric products like plastic bags and bottles cannot be dumped in sanitary landfills. These synthetic waste items are then combusted in the incinerators, which results in release of harmful gases and cause air pollution. Synthetic polymers can come in a variety of forms, such as common plastics, the nylon of a jacket, or the surface of a non-stick frying pan. But these human-made materials have a detrimental impact on ecosystems, which the United States National Institute of Health Researchers have called a rapidly increasing long-term threat. Understanding the ways that synthetic polymers degrade ecosystems is important in taking steps to eliminate this form of pollution. One of the most common environmental problems associated with synthetic polymers pollution is that 44% of seabird species are known to have ingested synthetic polymers that have been mistaken for food, according to the US National Institutes of Health, with millions dying from this ingestion every year. This broad-reaching death of shorebirds presents a significant environmental problem because shorebirds play a vital ecological role in maintaining the population sizes of fish and crustaceans. POPs, or persistent organic pollutants, are known toxins that remain in the environment for many years, such as the pesticides DDT and toxaphene. A 2007 study by researchers at the University of the Pacific sampled synthetic polymers found at coastal sites in the northern Pacific Ocean and found the presence of harmful toxins in every sample of synthetic polymers. These synthetic polymers can continuously secrete harmful chemicals into fish and wildlife when ingested and threatens the health of ocean fisheries that humans eat from. Beyond its evident pollution of oceans, synthetic polymers can also present environmental problems in the process of their production. The Environmental Working Group organization shows that the Viewpoint Chemical Company leaked contaminants used in the production of Teflon to local watersheds for several decades, according to the US Environmental Protection Agency. These chemical accumulates the grills of fish and can travel in high quantities up the food chain. Even beyond their persistence in oceans and water pollution from their production, synthetic polymers are a significant challenge on land because they are often disposed of in landfills where they will remain for centuries in the future, slowly leaching toxins into soil as time passes. According to the Clean Air Council organization, Americans alone use an estimated 102.1 billion plastic bags, synthetic polymer, each year, and less than 1% of these bags are recycled. Not only do these synthetic polymers slowly leach harmful chemicals in the soil, their longevity and non-biodegradability means new landfills will be a constant need as synthetic polymer use continues and grows. There are many other aspects of the polymers and related environmental issues. We all are eagerly waiting to listen from the Honorable Speaker, Professor Shunmugam. So, without wasting time further, I thank you all for your patience and announce the session open. Thank you.
Thank you, Principal Sir, for your very nice and informative inaugural address. May I now request our IQSC coordinator and also our present departmental head, Dr. Progamay Pal, to give his to deliver his welcome address for this webinar. Dr. Progamay Pal, please.
Thank you, Soumitra. Audible? Am I audible?
Yes.
Thank you, Soumitra. And also my colleagues of the Department of Chemistry for giving me the opportunity to deliver this welcome address. At first, a very good evening to all the participants. I am much glad to welcome you all to this webinar on my behalf, on behalf of the Department of Chemistry, and on behalf of our entire Fakirchand College family. As you all know, that today's webinar is organized by our Department of Chemistry in collaboration with the IQSC of the college. Today, our distinguished speaker is Professor Raja Shunmugam. We are really glad and honored to cordially welcome you, Professor Raja. A polymer is a substance or material consisting of very large molecules or macromolecules composed of many repeating subunits. Due to their broad spectrum of properties, both synthetic and natural polymers play essential roles in everyday life. Polymers range from familiar synthetic plastics such as polystyrene to natural biopolymers such as DNA and proteins that are fundamental to biological structure and function. Nowadays, synthetic polymers are used in almost all walks of life. Modern society would look very different without them. The spreading of polymer use is connected to their unique properties like low density, low cost, etc. For a given application, the properties of a polymer can be tuned or enhanced by combination with other materials as in composites. Their application allows to save energy, protect food and drinking water, save land and reduce use of fertilizers, preserve our materials, protect and save life, etc. But the non-biodegradable nature of synthetic polymers makes them a permanent waste. There are several serious environmental issues with polymers, which include air pollution, water pollution, occupational hazards, drug pollution, marine pollution, health hazards, etc., which have turned polymers from a hero to a villain. In the last decade, continuous research advances have been observed in the field of environmentally friendly polymers and polymer composites due to the dependence of polymers on fossil fuels and the sustainability issues related to plastic waste. Biopolymers are nowadays considered as among the most promising materials to solve these environmental problems. Blending currently represents a very cost-effective strategy to increase the ductility and impact resistance of biopolymers. There are many other explorations to minimize the environmental issues. So with these few words, and once again extending a very warm welcome to all of you in our present program, I am requesting my departmental colleague Dr. Soumitra Mondol to introduce our today's speaker. I know that all of you are eagerly waiting to hear from him. So Dr. Mondol, please.
Thank you, Prabhada. Now I am Dr. Soumitra Mondol, convener of this webinar. Happy to introduce today's webinar speaker, Professor Raja Shunmugam. Raja Shunmugam is now professor at Polymer Research Center and Center for Advanced Functional Materials, Department of Chemical Sciences, Indian Institute of Science Education and Research, IISER, Kolkata. Raja Shunmugam received his bachelor degree in 1994, a master's degree in 1996 from VOC College, Manonmaniam, Sundarnar University, Tamil Nadu. He received his PhD degree from the Indian Institute of Technology, Madras, under Professor R. Dhamodharan. He then joined Professor Gregory N. Tugh's laboratory in the Polymer Science and Engineering Department at the University of Massachusetts, Amherst, as a postdoctoral research associate from 2003 to August 2008. From September 2008 to 2013, he was an assistant professor in the Department of Chemical Sciences in IISER, Kolkata. From January 2014 to September 2018, he was an associate professor in the Department of Chemical Sciences at IISER, Kolkata. He was the head of the department from February 2014 to March 2016. From October 2018 onward, he has been a full professor in the Department of Chemical Sciences, IISER, Kolkata. He was a recipient of the Ramanujan Fellowship for the Department of Science and Technology, Government of India. He received the Joint Runner-Up Award in the 6th National Award for Technology Innovation under the Polymeric Materials category, 20th January 2016, for the arsenic sensing work. He also received the Joint Winner Award in the 7th National Award for Technology Innovation under the Polymeric Materials category, 1st March 2017, for developing a sensor for nerve agent. In 2018, his group received the Joint Runner-Up Award in the National Award for Technology Innovation under the Polymeric Materials category for developing new polymer for drug delivery application. Raja is the recipient of the coveted NASI Reliance Industries Platinum Jubilee Award for the year 2019. With these few words, I am honored to invite Professor Raja Shunmugam to deliver his valuable lecture. Professor Shunmugam, please.
Very good afternoon to everyone. Thank you, Dr. Mandal, for the very elaborated introduction. I must thank Head of the Department, Dr. Paul, for the kind invitation. And my heartfelt thanks to the Principal, Dr. Chanda, for the very nice introduction on polymers and its all pros and cons. Thank you so much, and it's a great honor and pleasure to meet you all and deliver my seminar and whatever the knowledge I have gained through these years, I would like to share it with you all. I hope I am audible, right?
Yes, sir.
Yes, sir. Yes, sir.
Yeah.
Yes.
Yeah. Let me share my slides. All right. This is, I hope these slides are visible. Yeah. The topic of this webinar is very interesting. Environmental hazards of polymers and its remedies. From the title, I can see that from my talk, environment is there, hazards are there, polymers are there, and of course remedies are there. Only of hazards of polymers is not there, okay? But you will definitely appreciate at the end of the seminar.
The slides are not visible.
I have not shared it. I'm just just giving my introductory remarks, and I will share it again. So still it is not visible, huh?
Not visible, sir.
Okay. Somehow it's, can you see it now?
No, sir.
What is the problem? Now? No. Can
No,
you
sir.
see?
No. It is not visible.
I really do not know what is up.
Excuse me, have you clicked present now?
I'm sorry?
Have you clicked present now?
Yeah, there are three options.
Present now, then share. Present, first you click present now, then share.
Present now in where? Actually, there is a sharing option. Normally, that
Right
only.
inside.
Yeah. Then there is.
Then you share.
You
click share. Click share.
Yeah, then it shares your entire screen or a window or a tab. Three options it is giving.
Yeah, entire window, then share.
The sharing is not happening. Now it is happening. Yes.
Now it is happening.
Can you see it now?
No, the slides are not visible.
Slides are not visible.
You press share entire screen.
Yeah.
Can you press
I did
share
that.
entire? You did that.
1 second.
Yeah, now it's visible.
Now it is visible.
Now it is visible?
Yes,
Yeah,
yes,
Yeah,
yes.
it is visible. Yeah.
Thank you so much. Sorry, I'm sorry about this. Somehow it is not, but I am glad that it is solved. Thank you so much. Yeah, as I mentioned, I'll be touching the environment, hazards, polymers, and remedies. I am sure you will appreciate the connectivity to the topic. I am also surprised to have a question even before the session from our esteemed audience, one of the audience. The question was, why don't we stop even using polymers? So on a lighter note, if we do that, people like me will lose a job. So, but definitely we will try to give an answer why we should not stop using plastic. That you will come to know. Of course, there are hazardous nature is there from the polymers, but we will try to capture whatever the best way to proceed with the polymers. So as I came to know from the audience, most of the students, participants also there. So I would like to start with few thoughts on science and research. So as you are young minds, my advice is you should be always look for the new results. Okay. Suppose if you are doing a project, even if it is a laboratory experiment, even though you have a procedure to carry out, suppose if you are taking a A and mixing B, you are supposed to get a C. That is a given procedure. You look for the new results and even observe what is happening. So that is the curiosity-driven observation at the laboratory level. If you are doing a research, that will take you to the next level. Okay. So some of the serendipity results, which is the greatest invention till date. For example, discovery of penicillin. Okay. When Alexander Fleming, his routine job is to check for the pile of petri dish for the growth of bacterial thing. That is his routine job. Suddenly, one fine day, he found that the petri dish doesn't have any growth of bacteria. He could have just thrown the dish and he would have moved on. But he didn't do that. He was looking for the reason why there was no bacterial growth. That is the invention of penicillin, and that is the best medicine so far, right? That is the accidental invention. The second one is very much related to polymers, and Dr. Plunkett, okay, he's a DuPont scientist. His routine job again to check the cylinders that containing perfluoroethylene gas, because that is he uses for the refrigerant gases. DuPont is known for making refrigerant gases. Okay, so Dr. Plunkett's routine job is to check the cylinder, and one fine day, the gas cylinder has white, solid, slippery powder. Okay, again, he did not move away by not noticing that white, slippery powder. He analyzed it. At that time, this is the greatest property, you know, like chemically inert, high melting point, and low surface tension. These three catchy property then evolved to develop the great Teflon. Okay, so that is the greatest polymer invention ever that happened by accident. So my advice is, please always look for new results. If that one chemical procedure is happening, it's fine. If it is not happening, you always ask question why it is not happening, then look for the reasons. That will take you to a next level. So these there are of course several serendipity results. These are not the only results, but I thought let me take two and give a small thought about the science. The second way of carrying out the research is patience and perseverance. For any goal, you need to have a patience and perseverance, particularly for the research. When you have a strong hope and plan and the perseverance, you will really reach the goal. Okay. The discovery of aluminum by Hall. When he was a student, class 9 student, the teacher was giving him a hint, saying that making aluminum is the toughest process. Somehow Hall took that in his mind. He was aiming to make aluminum in a cheapest way. That was his aim. Believe me, this is the best purifying process from the ores. The separation of aluminum from the ores. Hall's procedure is the best in the world so far. So that produces the cheapest purification method for aluminum, and still we are using that procedure. So that is the perseverance example. Okay. Then we all know about the Madame Curie's family, the four several Nobel Prize winning family. Okay. So again, that invention is an example for the sustained effort and perseverance. So my advice is you always look for the results, ask questions, and you should not lose your hope. Patience and perseverance are very, very important for particularly the students who choose research as the field. Another important to classification, which I would like to share it not only for the students, also for the younger colleagues. The science normally divided into two parts. So basic science and applied science. Okay, so normally even the departments will be divided like basic science, applied science. Basic science comes out from the core basics like biology, physics, chemistry kind of. Or when you do a research from these, the basic thing, it will be considered as a basic science. The word applied comes when you are having an application oriented or when you are merging with another subject, then the word applied. But the modern science moving towards the relevant science. Let me say what is a basic science. So basic science is when you study something, a new reactions, suppose if you are a famous organic reaction or a physics experiment, whatever it may be, you read it, you understand the theory, and you go to the lab and you take A and B, and you are mixing it, getting a C. So A plus B, A and B are already well documented, but the C is a new product which you are generating by understanding the theory and you are creating. Now you are analyzing the C and making the data. Definitely this is an addition to the knowledge addition to the A and B, which is already well documented, but you are adding the value of C to the literature and you are moving on. So that's how the basic science being developed. But what is the relevant science? Relevant science means you are making some molecule for what? When you put the question for what, then you are moving towards the relevance. Okay. Now, if you ask what is the relevance for the researchers at this present situation, COVID. Every funding agency, every country, and every research lab looks for a solution to a COVID condition, right? So the present situation and the present science, the relevant science, if you wish to do, and if you can develop a molecule that related to how you, suppose if you understand the COVID, or if you make a molecule that prevents the COVID, all type of research is relevant science. It's not only the COVID, of course, every part of the thing. Suppose if you are making a molecule related to cancer, So the point I wanted to register here is application oriented. Suppose if you are making a molecule, then if you ask the question for what, then the science becomes relevant. Relevant to whom? Relevant to the society, the users. So the present science is driven by the relevant question. Okay. So when you do a relevant science, obviously is a teamwork. A chemist will make the molecule with the support of the physicist to understand the theory behind. Then when it has to be applied to the life, then we need to collaborate with the biologists, doctors, to understand how it works in the cellular system, then animal, and then the human model, right? Then comes the that driven by the teamwork. So from my point of view, both basic science and relevant science are important. But if you really have a aspiration like getting a more funding, high impact publication and recognition, obviously you need to work on the relevant science. Then your chances of getting everything will be higher. Of course, this slide also related to the previous slide. When you do a relevant research, it needs a organizational behavior triggered by the needs of time and the social commitment to connect. That is very important. So these are the few things which I wish to register the young minds and also my colleagues. With that, I would like to move on to my seminar that is titled Functional Polymers for Heavy Metal Removal from Drinking Water. So before going to the actual topic, I want to register what are the problems in the environment, how these are all happening. So the source of water contamination has two sources, the point sources and non-point sources. For example, the localized sources like industrial process, mining activity, these will be producing the water contamination. The unidentified source, for example, the water discharges and runoff, these are all non-point sources. If you know the point sources, you can actually minimize the pollution. So that is the main point on these two slices. These are the three types of water contamination: the biological, where the infectious disease and oxygen demanding waste will contaminate water. Then from the chemical contamination, mostly the fertilizer, then toxic inorganic material, even the organic, like persistent organic pollutants. What is the physical contamination? It is due to the sedimentation, thermal pollution, and solid waste. I'll be briefly mentioning about all these contamination in the subsequent slides. First, let us see the biological contamination. The pathogenic microbe contamination, for example, the group of microbes infects through the drinking water and food. The group of microbes are mentioned here. Similarly, some viruses may grow in human alimentary canal or in the mouth and larynx. These are the examples for the virus contamination. Oxygen is a very important element, both in the water and air. So we need to keep the oxygen balance everywhere. Otherwise, the livingness will be questioned. The food waste, dead plant, and animal tissue that consumes oxygen in the dissolved water. Suppose water also should maintain a certain amount of oxygen, then only the livingness, and even while taking water also, it will be useful for the human. When these food waste and dead plant are consuming the amount of oxygen in the water, then the water becomes more contaminated, right? Then that water also not a good water. The chemical contamination happens, of course, from the fertilizers like phosphorus and nitrogen-containing fertilizers. Then comes the heavy metals like arsenic, enters in drinking water mostly in the form of inorganic trivalent arsenic 3 and pentavalent arsenic 5. That will create skin cancer and more damages to the thing. The persistent oxygens like halogenated organic compounds and these halogenated organic compound has a high lipid solubility that will interact with our living systems. So, this is again the persistent oxygen pollutants also very dangerous for the contaminant in the drinking water. What are the physical contamination? The sediment contamination. High levels of sedimentation in rivers leads to the physical disruption. When there is a degradation of water quality by any process of that changes water, then the livings in the, for example, fishes and other living animals in the water gets disturbed due to the change in the temperature. That indirectly influences the living cycle. Then comes the most important solid waste contamination, where while giving the introductory lecture, the principal sir and the head of the department, and Dr. Paul, everybody touched about this topic. I also would like to spend a few minutes in this particular slide. Here comes the importance. So, banning, we always see the slogan, ban plastic. But as a polymer chemist, that slogan always hurts me, because can we imagine a life without a polymer in a day-to-day life, from coming out of the bed to going back to the bed? So we have to, from the toothbrush to the eating, and then comes your travel, scooter, car, whatever the means, you try to travel from one place to another, cycle, tube, everything. Then comes to your medical emergencies like heart valve, artificial nerve, operational instrument, everything, and contact lens, even your heart valves are made of biodegradable polymers. You cannot imagine a life without a polymer. We cannot just like that stop using polymer. We cannot. If we have to use it, then we need to go back to our olden days cave life. Then only we can avoid plastics. For a modern life, we are used to have this life right now. Right now, going back to that cave life is impossible. So stop using the polymer is impossible. How can we then stop the contaminant? So what we have to do, we need to read this slogan. You stop using the single use of plastics. What is single use of plastic? The carry bags. The problem is not about biodegradability. Problem is with the users. Olden days, suppose 15 years back, if we consume a cool drinks, the cool drinks will come in a bottle. We consume the cool drinks and we give the bottle back to the shopkeeper. Now, what happens? All the plastic bottles, everything comes. So we carry the cool drinks or whatever the food available, we carry out, come out of the places, we consume it and throw it in the environment. Even the bottles are not biodegradable. We cannot recycle the bottles. Early days, we used to have the bottles and metal containers. We used to give it back to the shopkeeper. Similarly, it is, again, I'm stressing the point, bottles or metals are not biodegradable. Still, you always used to have those. Similarly, plastic also not biodegradable, it is fine, but we should stop using the single usage of the plastics so that the accumulation in the environment can be stopped. So the problem is with the users, not with the material. That's exactly I want to mention. Of course, if we, a polymer chemist like me and other colleagues, if we can work on the biodegradability and if we can crack this carbon-carbon single bond and we can recycle it, then that is the best solution to the contamination. However, we can reduce the use of the polymer, but we cannot stop using the polymer because it is there already in our life. So we should stop using the single usage plastic. That exactly I wanted to emphasize. I hope you all will also appreciate that point. So with that, let me slowly move on to the change my gear to the actual topic. In my presentation, the next all my presentation, I will be concentrating on how arsenic contamination is happening and how the remedies can be done. That is going to be my next few minutes topic. This is the entire region of arsenic contamination in India. So if you can notice, it is always Ganges, but so these are the regions where the fully contaminated arsenic 3 area. And I work here in the West Bengal, so I'm very much concerned about this when I started my career. So I wanted to develop this mechanism how to detect the arsenic 3 and how to separate arsenic 3 from the drinking water. That was my aim when I joined IISER Kolkata in 2008. So while studying the arsenic 3 binding in human protein, how it happens, we learned it that the thiol group SH with the CH2 that nicely binds with arsenic 3 system. That's how it enters to the living systems. The arsenic 3 binding protein has the CH2SH. CH2SH, that nodal point is very, very important for arsenic 3 binding. Then it easily gets into the living system. That is very well documented in the literature. As a chemist, I was very much excited about that CH2SH bond so that I can also capture how to bind the arsenic 3. That was the learning lesson as a chemist. Okay? Then we also came to know, I must appreciate my student Saurav for this excellent equation. While searching for the arsenic 3 literature, he found this interesting equation. Arsenic 3 gets oxidized to arsenic 5 in the presence of potassium iodide and dilute HCl, acidic condition. This reaction is very specific for arsenic 3. It binds only, this happens only to the arsenic 3. Arsenic 3 gets oxidized to arsenic 5 in the presence of KOI3 and it liberates the iodine. Okay, this is an excellent, very unique reaction for arsenic 3. It happens only with arsenic 3 and it liberates iodine. In my mind, we all know that this starch iodide experiment, which is a class 12 experiment, it's beautiful experiment to trap the iodine. Even very trace amount of iodine can be detected using this starch iodine. Even though the mechanism is not still clear, but it is a very excellent tool to capture the iodine. Okay? So these two reactions were in my mind for a quite long time. Then I must also register here that there are several other heavy metals like mercury, cadmium, zinc, lead, and in the literature for the detection or sensing these heavy metals, there are a number of reports, but there is very few direct chemical method that can detect arsenic 3 because of the atomic nature of arsenic 3, which is very, very difficult to create a molecule that binds arsenic 3 directly. So that is why the arsenic 3 sensor or detectors chemically is very poor in the literature. So keeping all these factors in my mind, we try to capture this particular reaction, arsenic 3 getting oxidized to arsenic 5 in the presence of AO3 and liberating iodine. We want to link these two, chemistry and that starch iodine experiment. We came up with this interesting molecule. This left-hand side is a monomer and the right-hand side is the polymer version of this monomer. This particular molecule is called norbornene and this is rhodamine functionality. We linked rhodamine to the norbornene and this is a specially modified cyclic point where the iodine and the acid can play a role to detect the liberated iodine. So that's how we designed to trap the iodine released in the oxidation of arsenic 3 to arsenic 5. So before going to the detail, a little bit of background on what are polymers. Those who have not studied polymers yet, I just want to give a few introduction. Poly means many and mer means the units. So combination of small molecules. Into the left hand side is example for monomers. When you join them covalently, and it forms a very nice chain. So you can control the growth of the chain by controlling the initiator, temperature, and chemical, all the factors. By controlling them, you can actually control the growth of the polymer chain. Okay, but a polymer is a combination of small molecules, so combination of the monomers in a single chain, that is called polymers. Okay? And types of polymers, when you start with a single monomer, say for example styrene is a monomer, when you polymerize free radically, it forms polystyrene. So if you start from one monomer and you grow the chain and get the polymer, then that particular polymer is called homopolymer. When you are using two different monomers, say for example styrene and the methyl methacrylate, these are two monomers, and if you are polymerizing them and allowing them to grow in a single chain, as you can see, the red dots are, let's say, methacrylate, and the blue dots are, let's say, it's styrene. Then the grown-up chain is called a copolymer of styrene and methyl methacrylate. So in that chain, you have both monomer units present in the chain. That is called the copolymer. And the copolymers are having two different classifications, block and random. So block copolymer has a sequential growth. First, you polymerize the first monomer, let's say it's methacrylate, to some extent, then stop the polymer, you add the second monomer, then allow the chain to grow. In that way, you grow the complete chain. And if you take the single chain, the first sequence to some extent will be the methacrylate. The second sequence will be to some extent will be the styrene. That is called the block copolymer. Then if you see the randomness, if the positions are not really defined, it will be randomly positioned, like acrylate, styrene, then few acrylates, then few styrene. Then that particular chain is called the random or statistical copolymer. These are the type of polymer chains that I thought it will be useful for you to understand better. Now I am coming back to our design. As I said, this is the monomer and this is the polymer, represented the chain as N. Then the chain, if you imagine this molecule is having 20, then each 20 molecule will carry the individual rhodomine, and that's how the polymer chain goes. Now coming to, let me show the results. This is the absorbance UV data, and from that we have created the calibration curve. And having the sensor molecule or the directing molecule in the polymer is, you can make a paper strips like a pH testing paper. You might have all used the pH testing paper, the litmus test, where you want to check the what is the pH level after the endpoint and before the endpoint, right? By seeing the color. So that was the idea of making paper strips out of our polymer, so that you just take the paper strip, dip it in an unknown solution. If arsenic is there, then you will see a color change. So that is that shall be used for the in-field. You don't have to go collect the sample and go to the laboratory. You take the paper strip, go to any water body, dip. If you see the color change, then you can say, hey, there is a arsenic contamination. Like that you can say. That was our idea of developing that paper strip, and this is exactly how we demonstrated the in-field sensing. And we just went to the nearby pond and we dipped the paper strip. You can see the color change, right? So from the colorless to color change, that itself clearly mentioned the presence of arsenic 3. Well, we have of course developed arsenic 3 sensing method, but are we the first one to sense or say that arsenic contamination in West Bengal? No. There is several research labs are doing that, and it is the arsenic contamination in the Bengal, particularly the Ganges body, is very well documented, and it is very much there in the literature. So what we have done is a quick and reliable paper strip to detect arsenic 3% in the drinking water. Well, our job is not yet done. as I said, we all know that arsenic 3 presence is there in the many of the surrounding West Bengal state. Now, after showing that arsenic is there, it is our responsibility to remove, give a solution for the removal. So towards that motivation, we developed a polymer-based resins that can trap arsenic 3 from the drinking water. That was our second motive. Then we prepared the polymer and put it in a small column, and we demonstrated that by several cycles you can trap arsenic 3 if you allow the water to pass through the column. Then you collect the water. When we check the arsenic presence, it is almost completely removed. So we were excited about that result, and we published that work. Then I must start the second story. I also must thank ADO, a plasticizer developing company in Kolkata-based company. I must thank the managing director, Mr. Sanjeev Pariyal, who got excited about the previous results about the arsenic sensor and developing, and particularly the trapping system. He requested us to develop a system for their company so that they can develop and take the molecule to the product. That excited us a lot. Then I involved 3 of my students, Rajan Kumar, Tapandu, and Pawan Kumar. They reworked on these molecule and they developed the first sensor molecule of this type. And these are the sensing behavior from the spectroscopy analysis, and you can see that from this table, it is very specific colorimetric response gives only to the arsenic 3, while other metal ions could not give any response. Then, like the previous model, we also developed a paper strip model that changes the color from one colorless to color. That's how we demonstrated. And we also demonstrated the in-field sensing capabilities of these paper strips. And we demonstrated the color change. Then what we did, this is exactly the IISER Kolkata location. It is located in the Nadia district. Then the ADO company, ADO additive is located in the Madhyam Gram. It is about nearly 40 kilometers between the IISER Kolkata and ADO additives. What we did, we collected water samples from the water bodies between these two areas. And then we tested the arsenic presence using our arsenic sensor. And these are the data near the Biroi and Aispur. And you can see that these are the arsenic 3 contamination in the water body. Okay, and similarly, the Uttapara and Tezpur, Tetulpara data also presented here. All from all this data, you can see that. I must also tell here, the World Health Organization allowed arsenic 3 level in drinking water is 10 parts per billion. But what we see is all in parts per million. It's a high amount. So it's really a concern. So we really wanted to make a trap for the arsenic 3. So that's how we worked on our polymer and we created a trapping system. This is a general, even the AquaGuard, Kent, all water separating system will have all these 3, the green, red, and black. All these 3 are common in your water purifying system. In addition, what we have done, this cartridge, suppose if we keep the polymer that we have synthesized next to these columns, it will remove the arsenic 3 completely. That is the finest invention from us. And I must again thank additives. So after putting that polymer column to the water purification system, these are the data where we can completely trap arsenic 3, and there are several cycles we have tested. And after testing that, we even launched the product. I must thank Sanjeev Pariyal, Additive MD. I must also thank former Secretary Dr. T. Ramasamy sir. Then this is our director, Professor Saurabh. We are always grateful to them for launching our product, both polymer separation column as well as the sensor molecule. He gave us a lot of awards and recognitions. And then I also would like to show the products developed from our laboratory. These are the sensor for cadmium, nerve gas agent, iron, and arsenic 3. These are the other sensing molecules that have been developed in our laboratory, which also invited a lot of awards. And most important, prestigious is the academies, National Academy and Reliance Platinum Jubilee Award for the innovation in the 2019. That is very important. And I also would like to show the research theme. So far I was mentioning about sensing the heavy metal as well as poisonous gas. And we are also interested in making polymers for drug delivery, particularly cancer theory. In next few minutes, I would like to quickly go through this topic also, so that it will give you overall overview of the work that we are doing. The normal cell versus cancer cell properties are developed. As you can see, chemically, these cancer cells are much different from normal cell. For example, slightly acidic in nature, and some enzymes will be overexpressed. For example, folate, biotin, and some receptors will be overexpressed. As a chemist, we always try to capture those additional chemically rich environment in the cancer cell, and so that we can fine-tune the drug delivery aspects. And in the drug delivery, there are two passages: the passive drug delivery, where enhanced permeability and retention EPR effect, and that can be used. Whereas in the active drug delivery, you can always target the nutrients like folate, biotin, transferrin, and glucose, antibodies and peptides, and receptor mediated enzymes. So these are the two way of targeting the cancer cell. And this is the progress of the field, how the drug delivery in the cancer evolved. It is up to 2008. We are now in 2021. It all started in 1970 from the simple ribosome-based polymer. Then the targeting group, nanoparticle, multi-arm, nanotube, carbon sheets, graphite, everything has come and evolved. The field is evolved till 2021. Despite the evolvement, in my personal view, the amount of drug reaching the cancer cell and the drug without harming the normal cell, these are the huge challenging in this field. So with keeping that in our mind, we designed first with amphiphilic polymer that forms vesicular kind of, that the cartoon represents two types of vesicle, vesicle and reverse vesicle. When you change the solvent polarity from polar to non-polar, the vesicular, the surface molecule reverses it. That's why it is called reverse vesicle. Okay. The vesicle differs from micelle. Micelles are smaller structures with a dense core, whereas the vesicular structures are the bilayer structures with the halo in the middle, where you can encapsulate the drug molecule and release it in the cancer site. That is the idea of making this vesicle. And we developed these molecules, and we also successfully demonstrated its application in the cancer therapy. And that was our first model. Then we also fine-tuned these vesicle structure, and this is the contribution of making the vesicular structure and deliver this drug molecule in the cancer cell. Whatever the way we deliver using a vesicular structure is non-covalent approach, because we create a carrier structure, then we incubate the drug molecule, then we release it by disruption of these aggregates in the cancer site, and immediately we release. So the drugs molecules are not covalently bound to these polymer chains. We are just incubating into the interior of these structures, and we are releasing. That's why it is called non-covalent approach. Whereas we have also developed the polymer where we have attached the drug molecule through the chemically stimuli linkage. For example, these hydrazone linkers are very sensitive to the acid. We link the doxorubicin, which is a cancer drug, to the polymer backbone using hydrogen linker. And we also employed this polyethylene glycol, which is necessary for the polymer chain to be biocompatible. Okay, so that's why we employed this polyethylene glycol into the polymer. Now, because of this chemically rich amphilicity, when it is given into the biological environment, for example, aqueous conditions, due to the polarity, it forms aggregates, and the polyethylene glycol functionality will be outside, and the relatively hydrophobic moieties will be inside. That way, the drugs will be protected. So that's started this covalent system. Then we fine-tuned by incorporating the site specificity to the nanocarrier by adding the folate, because folate receptors are overexpressed in the cancer cell. That's why we incorporated folate, so that the nanograde with the folate outside can be identified by the folate receptor. Then the nanocarrier can easily internalize into the cancer cell. That was the idea, having this model. Then, so far we have the idea how it is going into the cell. But after going into the cancer cell, what is happening? That information we do not have. To track the nanocarrier path inside the cancer cell, we wanted to include mitochondria targeting functionality. So, which is a TPP functionality, triphenyl. So, TPP has a positively charged. So, mitochondria will have a negative surface, so that will attract the TPP functionality. That is the logic behind in incorporating TPP functionality. Now we have designed the TPP functionality along with biotin and the drug molecule. The biotin functionality will help to get into the internalization of the cancer cell using through the biotin receptor. After getting into the cell, the TPP functionality will take care of the nanocarrier directly going to the mitochondria. Why we have to target mitochondria? Because that is the energy source for the cell. If you target and attack the mitochondria, stop functioning, then cell growth will be stopped. That's why we have to target mitochondria. Okay, so this is the latest design. With that, we are very much happy and successfully demonstrated that our nanocarrier kills cancer cells very efficiently, and that invention also got the innovation award in the 2019. With that, I would like to conclude my presentation by showing this interesting slide. In the sensor field, we are successful bringing our results into the market, thanks to Ado Additives for encouraging our result to bring it to the product. Similarly, our drug delivery work also, we have made a few polymers. We also tested at the cellular level. We need to go cross this line with the help of clinicians and biological laboratories. We are hopeful that we will surely cross this line one day and get into the market. With that, I would like to thank my bright students. Without them, it is very impossible to carry out all this work carried out by all my young team. They are very vibrant and energetic. I must also thank IISER Kolkata for everything, and DST, DRDO, CSIR, CARS, NPO, DRDO, and Ado Additives, particularly for bringing our research into the market. And thank you all for your kind attention. Thank you very much.
Thank you, Professor Raja Shunmugam, for your very nice and informative presentation. So now the session is open for discussion. So if anyone has any question, okay, he or she can ask our resource person, Professor Raja Shunmugam. So please give your question. You can ask here. Have anyone any question? Then he or she can ask freely.
Yeah, please feel to.
So I think there is no questions right now. So we are at the fag end of our program. Now I am requesting my departmental colleague, Dr. Moumita Sen Sharma, who is also an associate professor of this department, to deliver the formal vote of thanks.
Yeah, thank you, Dr. Pal. Good evening, everyone. I hope I'm audible. Well, I have been entrusted with the responsibility of conveying the vote of thanks, and I feel greatly obliged for the honor. So, firstly, I would like to thank our distinguished speaker, Professor Raja Shunmugam, for delivering such an enriching talk. I hope we would be fortunate enough to hear you again in the future, sir. I would like to thank our principal, sir, Dr. Shomen Chando, for his constant support and encouragement. I would like to thank our IQSC coordinator, Dr. Programay Pal, and each and every member of our college, including those in college administration, for extending their wholehearted cooperation for execution of today's webinar. Last but not the least, I would like to thank all the participants without whose involvement this program wouldn't have been a success. Take care and stay safe. Thank you all.